Hi, I'm Sangeetha Nair, and this is easy. I don't have any disclosures other than the fact that I'm a Hologic employee. So as SARS-CoV-2 is rampaging across the world, there's something to be said for CMV. It's already infected most of the world's population, with infection rates approaching 100% in many different geographies. But the silver lining here is that CMV mostly establishes latent infection and does not cause severe problems usually in people with a normal immune system. However, because of its ubiquitous nature, it is the most common opportunistic pathogen in people with organ transplants, and also causes issues with people with many different immunodeficiency conditions. And what's really intriguing is that it is the most common infectious cause of birth defects in babies, causing all sorts of problems ranging from vision loss to hearing loss and developmental delays. Nucleic acid testing is the cornerstone for both diagnosis and management of CMV infection. So today I'll just be walking you through the various aspects of the Aptima CMV Quant, especially the analytical validation, and ranging from the intended use to the performance of this test. So this is a quantitative test that is intended for quantification of CMV DNA from plasma samples for the management of organ transplant patients, both solid organ as well as stem cell transplant patients. And as you've seen from the third international consensus guideline, there's no threshold for triggering treatment for CMV patients. And in the absence of that, serial monitoring of CMV viral load is the way for managing such patients. For Aptima CMV Quant, the assay is composed of a reagent kit of assay-specific reagents, as well as a box of controls. And one unique feature is that we also have a box of calibrators. It's just one single calibrator that is run on a regular basis. And what this does is ensures accuracy, especially at the low end of the assay range. And this is a special feature of this product, and because oftentimes we make so many medical decisions for these viral load assays based on the concentration of virus at low concentrations. This assay is run on the Panther platform, which automates every step of the process, from nucleic acid extraction to amplification, detection, analysis of results, and communication to the LIS. What is good about the Panther is its small footprint compared in combination with the high throughput that this platform offers. And it reports results in real time, as well as being a smart instrument. It has a lot of sensors and process controls, which signals the users in case of errors, and can track consumables and assay fluids, which are available on board the Panther. And it also has complete random access, so bye-bye to batching. So you can load the samples as they arrive in your lab and test multiple assays and multiple sample types at the same time. And another great feature is the ability to test primary tubes directly on the Panther platform. And you know how important that is for traceability of results, right, from patient to results. That is provided by being able to load primary tubes. Now, when you talk about the workflow on Panther, you can collect whole blood using either EDTA tubes or the plasma preparation tubes. And these tubes can be centrifuged, and the same primary tube with the blood pellet at the bottom and plasma on top can be loaded onto Panther for testing. And if you so wish, you can also transfer it to the secondary tube. And the benefit of using the Hologic secondary tube is that it permits you to use as little as 700 microliters per test for running on the Panther platform. We've also done a lot of studies with alternate sample types, like saliva, urine, whole blood, and all of them have pretty simple workflows, but none of those are validated in the US. And we use them only for research use. Talking about assay design, this assay targets the UL56 gene of CMV. And I know the first thing that comes to mind is there are publications on drug-resistant mutations to the drug letermovir targeting to this gene. And right off the bat, let me tell you, the target region for this product is more than 500 bases away from the region from where any drug-resistant mutations have been reported. And this assay also has really long primers, and that enables this technology to be really robust in the presence of mutations, enabling equivalent quantification, even if mutations are present. And I know you heard from Karen's talk and also about the number of publications out there on the correlation between amplicon size and quantitation in various quant assays. So in this assay, the amplicon size is around 110 base pairs, and it gives results which are very comparable to that of many of the commercial tests, including the Roche Cobas CMV test, the Abbott REALTIME, and even the QIAGEN ARTIS. This assay is calibrated to the WHO standards, and the limit of detection and limit of quantification studies were performed with dilutions of the WHO standard in plasma and tested with multiple reagent lots. And we also confirmed the limit of detection and quantification with samples of various CMV genotypes as well as drug-resistant mutants. And as you can see from the last row here, the LOD and LOQ are 41 and 53 IU per ml, which is very much in the same ballpark as that seen for other highly sensitive CMV quant assays. Now, one thing to note is these results are with the WHO standards, while those presented by Dr. Lima as well as Dr. Harrington were with dilutions of the exact panel. And it's great to see such good agreement in LOD and LOQ at different sites with different panels, and yet very similar results. This assay also has very good specificity. We tested nearly 400 individual donor specimens, and the specificity of this test was really high, and we did not see any interference in the presence of potential interfering substances, whether they be endogenous or exogenous. And by that I mean endogenous substances like protein or cholesterol that may be present in the sample, or even human DNA, as well as things like drugs that may be present. And we do a lot of testing also with organisms that may be present in the sample type, bloodborne pathogens, other herpes viruses, or pretty much any organism that's likely to be present in the sample. And we take it one step further by pretty much testing all the analytes that can be tested on Panther in our cross-reactivity studies, and we always demonstrate that there's no cross-reaction with any of these agents. Being a quant assay, I have to put up something on linearity, and here you go. On the x-axis here are the target concentrations, and y-axis are the recovered results for Aptima CMV Quant with all 4 genotypes of CMV. And from the R-square values, which are about 0.999, you can see results are really linear across the complete assay range for all genotypes of CMV. Now to the big question: precision, reproducibility, and commutability, because that's something that's often talked about for CMV. So we did this in many different ways. The first step was to test a panel across the assay range, and it was tested with 3 different reagent lots on 3 different Panthers, multiple operators over 20 days. And this testing was done at one site. And as you can see, the contribution to variability from reagent lots or instruments or operators or any such thing is really minimal. And the maximum variability, when you put all these factors together, was 0.18 standard deviation log IU per ml across the whole assay range. And this is for testing with one site. that is at our San Diego site that you can see right here. We took it further and tested it at 3 external sites and pretty much got spot on the same results, with maximum variability still being at 0.18 standard deviation log IU per ml. Now, to take it a step further, it's the same panel that was used by Dr. Harrington's team for testing at the external site, and they also got the same results with this panel, and we've also rolled it out across various sites in Europe to get the same results. And now, we talked about precision of results and within a lab, and reproducibility of Aptima results across multiple labs. What about commutability of results between Aptima and different tests? In the talks by our previous speakers, we already heard about commutability with Abbott from Dr. Harrington's talk, and Dr. Lima told us about comparisons with ELITech LDT assays. So here I am with the COBUS CMV test. And here we have the Roche results on the x-axis and Aptima here on the y-axis with Deming regression results, which approach a slope, the slope of 0.98, fairly close to 1. Really good agreement and very minimal bias between these two assays. We also did a bland Altman plot. As you can see here, mostly all the samples recovered within half a log. And it should be noted that we did this testing with 275 specimens, and there were only 3% of specimens that were outside the plus or minus half a log. And even those, the maximum difference we saw was 0.67 logs. Again, when you think about the international guidelines, they talk about half a log as a significant difference when testing one replicate. So it's great to see that Aptima results are really commutable with those in the COBUS CMV test. We had a smaller data set in comparison of Aptima with the Abbott REALTIME. And here you'll see the slopes of 1, which shows really good agreement between these two tests. And the bland Altman plot, again showing that most of the results are within plus or minus half a log when comparing to Abbott. There are a couple here or there, but again, the discordant rate between them is like 3%. And I know there's a lot of talk about commutability of CMV, but what this data demonstrates is that if you have commercially approved tests, the results between them are really commutable, that you may be able to use these tests almost interchangeably without the need for baselining in each test. We took it one step further to look at commutability. So looking at clinical samples is one approach. Wider net to look at the same thing is by using the CAP panels. And we tested the 2020 CAP panels, which give you results in many different tests, ranging from Abbott in green here, and there's QIAGEN, Roche COBAS TaqMan, COBAS CMV, and Aptima in blue here. And as you can see, mostly all the results in all these assays were generally within half a log, with best agreement seen between Aptima results and the Roche COBAS CMV test. And just like we do CAP testing in the US, the common panels to use in Europe is the EQA panels from QCMD, that is for External Quality Assurance Program in Europe. And here also the results from testing in Aptima were very much in line with the consensus result that is reported by QCMD EQA program. And we also did one more analysis, and that is for the drug-resistant panels that are provided by QCMD. For this, the Aptima results again in blue, and that reported by the vendor in green. We saw very good agreement between the expected concentration and Aptima, showing that this assay is really robust, even in testing drug-resistant mutants. You saw a lot of data on SERACARE panels from our previous speakers. we also tested a couple of panels in R&D. That is the AccuSpan panels from SERACARE, as well as the AccuTrack panel. So one is a linearity panel, the other is a qualification panel, and pretty much the green is the value from the vendor, and blue is Aptima. The SERACARE results are from the Roche-Cobas-Stackman assay, and we saw fairly good agreement even between Aptima CMV and Roche-Cobas-Stackman. The main difference being the difference in sensitivity, because some of the low concentration panels, we did not get a result for the Roche-Cobas-Stackman, while we were able to get results for Aptima CMV. To summarize, the Aptima CMV Quant test is highly sensitive, as well as specific, with good linearity across the assay range, excellent precision and reproducibility of results within lab as well as between labs, and results being commutable with that of commonly available commercial tests, including COBA CMV as well as ABBOTT REALTIME. The reagents have 2 years stability, and you can pretty much leave the reagents on board for at least 4 days so that you can load the samples as they come into the lab and really practice random access without the need for any batching. And obviously, the ability to load primary tubes is a big one. The small footprint and the high throughput of Panther, as well as random access, also are great ease of use characteristics of this platform. So the good performance of Aptima CMV Quant assay in combination with the complete automation provided by Panther makes it a good option for CMV testing for any lab. And we have teams in pretty much every department across Hologic that work on development and launch of our products. This is just a photo of my team members in R&D who worked on this day in and day out for multiple years to get this product to the stage it is now, very close to launch. Thank you for your attention, and thank you for being here with us this afternoon. We enjoyed having you here, and have a wonderful rest of your day.